organisms, even the largest, begin their life from a single cell known as the zygote. The zygote then grows and divides into two, wherein each parent cell forms two daughter cells. These daughter cells are mature and further divide to form new cells. These cells continue the division until an organism is formed. This process of cell division is vital to all the living organisms. During this process, cell growth takes place, followed by the DNA replication and followed by the nuclear division, finally leading to the division of the entire cell. However, it requires that the cell divides in a coordinated manner. This coordinated manner ensures that the cell divides correctly and the genetic material in the daughter cells remain intact. The series of events by which a cell duplicates its genetic material, synthesizes its constituents and eventually divides to form two new daughter cells is known as the cell cycle. All the cells in our body generally reproduces by cell cycle. Although the process of cell growth is a continuous one, the DNA synthesis takes place only during one particular stage in the cell cycle known as the S phase. After the DNA is synthesized, the replicated chromosomes or the DNA are distributed to the daughter nuclei through a series of complex genetically controlled events during cell division. The duration of the cell cycle varies from one organism to another. A typical eukaryotic cell, like those in case of the human beings, divides once in about every 24 hours. Whereas in case of yeast, the division takes place in about 90 minutes. Also, the cell cycle is divided into two basic phases, the interphase and the M phase or the mitosis phase. In case of the interphase, it is the resting phase during which cell prepares to divide by undergoing cell growth and DNA replication in a sequential manner. The interphase is subdivided into three phases: the G1 phase or the gap 1 phase, the S phase or the synthesis phase, the G2 phase or the gap 2 phase. In the G1 phase or the gap 1 phase, it is also known as the first growth phase. Here, normal cell functions occur and the cell continues to grow. It remains metabolically active, but the DNA does not replicate. In the S phase or the synthesis phase, the DNA synthesizes or replicates and the amount of DNA in the cell doubles without any increase in the number of chromosomes. Also, the centriole in the cytoplasm begins to duplicate in the S phase. Finally, in the G2 phase or the gap 2 phase, the cell prepares for mitosis and cell division by synthesizing proteins while the cell still continues to grow. The next phase of the cell cycle is the M phase, also known as the mitotic phase, where the actual cell division takes place. In case of the mitosis phase, it begins with the division of the nucleus and hence it is also known as karyokinesis. The M phase ends with the division of the cytoplasm, which is known as cytokinesis. Do you know that in the 24-hour average cell cycle of a human cell, proper cell division lasts for only about an hour, while more than 95% of the cell cycle duration is about the interphase? In adults, for example, in humans, some cells like those in the heart do not appear to display cell division. Animals also have many cells that divide occasionally, only to replace the cells due to the cell death or injury. So thus, such cells do not display further cell division and they do not divide regularly. So that type of cells exit the G1 phase and enter the phase called G0 phase or the quiescent phase. Here, in this stage, the cell remains metabolically active but proliferate only when the need means they divide only when needed. Also, in animal cells, mitotic division occurs only in diploid cells, whereas in case of plants, it is seen in both the haploid and diploid cells. In case of the living organisms, the cell cycle is a series of events that leads to the duplication of the cell and division, resulting in the growth of organisms. Mitosis or karyokinesis along with the cytokinesis defines the mitosis phase of the cell cycle.